Hello, my name is Cameron. I'm going to introduce fluorescence microscopy and computational processing and analysis of biological images. Fluorescence microscopy is a very useful technology in cell biology. The special thing about fluorescence microscopy is that glowing labels emphasize the subject against the background, increasing the resolution of the image. For example, this image represents mammalian fibroblasts with multiple fluorescent labels. Compare this vivid image to this relatively bland image. This image represents mammalian fibroblasts without fluorescence. The cells are similar, but the microscopic technology makes a big difference in the image. In the fluorescent image, three separate fluorophores label distinct parts of the cells. The blue fluorophore labels deoxyribonucleic acid, or DNA, in the cell's nucleus. The magenta fluorophore labels actin proteins that form the cell's cytoskeleton. The green fluorophore labels vinculin proteins that help attach the cytoskeleton to the cell membrane. There are many fluorophores that are useful in fluorescence microscopy, but green fluorescent protein deserves mention. Green fluorescent protein, or GFP, is a natural protein that originates in the bioluminescent jellyfish Echoria victoria. Here is an image of Echoria. Since its discovery, scientists have engineered many mutants of GFP for use as tools in biotechnology. Since GFP is a protein, it is possible to introduce its gene to a cell so that the cell expresses its own fluorescent label. Fluorophores, like GFP, emit light when light of greater energy excites them. For example, GFP emits green light when blue light excites it. A fluorescence microscope shines light of a specific color on the subject to excite it. Simultaneously, its camera measures the intensity of the fluorescent light of a specific color that the subject emits. An image from fluorescence microscopy is a measurement with physical significance. It provides a lot of information. Greater fluorescent intensity at a specific point in an image means that more of the fluorophore was present at that position in the subject. If more fluorophore was present, then there was also more of whatever it was labeling. So, there's a correlation between the image's intensity and the abundance of very tiny details of the subject. Let's consider an example. This set of images represents an experiment in human osteosarcoma U2OS cells. In these cells, the blue fluorophore labels DNA in the cell's nucleus, and the green fluorophore, GFP, labels a protein named forkhead. Forkhead is a transcription factor, meaning that it associates with DNA. Normally, forkhead moves freely between the nucleus and cytosol of the cell, as the image on the left shows. Treating cells with either of two drugs, wart-mannan or LY294002, causes forkhead to accumulate in the nucleus, as the image on the right shows. We can look at the two images and recognize a difference, but the images also provide even more information. Here is one of the images from the set. We can see the blue nuclei within the green cells. We can see the positions, sizes, and shapes of these cells. We can see when the amount of green signal decreases in the cytosol and increases in the nucleus. All of these observations are examples of a qualitative analysis of the image. But we can do better than that. Digital images are visual representations of arrays of numbers. As such, they offer quantitative information. Here is the same image with outlines of the nucleus and cell. Computational algorithms can recognize particular regions like these in images. This process is called image segmentation. With these segmentations, we can computationally measure characteristics such as the count of cells, the area of cells, the shape of cells, and the total intensity of the green fluorophore in the nucleus and cytosol. These measurements are examples of a quantitative analysis of the image. Furthermore, we can perform these processes and analyses automatically on a large number of images to provide statistical power. Quantitative analysis and automation are the two great advantages of computational image processing and analysis. Together, they make a lot of information available from biological images.